The impact of the study was showing that by making novel combinations between various barley lines, we could improve some final traits of barley grain that are really important for the brewing industry. Well, it's actually something we were able to do because of lots of research that has been going on for decades. So there were studies set up to understand what genes were controlling a certain trait in barley called row type. So row type, I'll just use these are heads of barley or barley spikes. Row type describes how many grain are present on one of these heads or spikes of barley. And so this is what you see in the field waving at you in the wind at the end of the barley life cycle. And what I'm showing you here is kind of the wild version, or this is what barley before it was domesticated looked like. And this is called 2-row. Can you see that there are 2 rows of grain? There's 1 row here on this side, and then there's on the absolute opposite, there's another row, 100. And this is called a 6-row barley spike. And it's called 6 because if you were to look down kind of direct, like bird's eye view this way, I suppose, or this way, you'd actually have 6 rows. So rather than the 2 that you have here, you have 6. And that's because you have these additional grains forming because you have flowers that remain fertile. So this means that many farmers will not grow 6-row varieties because they can't sell that grain onward. And so it's used mostly for animal feed in the UK context. Now, animal feed isn't as, you don't get such a great amount of money for animal feed as you will for malt. So if you grow for malt, it's a kind of a premium barley product. Okay, so there's a lot of interest understanding, well, what's the genetic difference between these 2 varieties, and can we use that knowledge to maybe improve some of these 6-row varieties. So that's work that has been going on for 50 or 60 years. And so from that huge amount of work, we're able to narrow down 4 or 5 major genes that can control this trait. Basically, you can switch a barley from this, looking like this, to looking like this by altering 1 gene. So there's, as I said, there's 4 or 5 of those genes where you have that are that powerful at controlling this trait. So in the 6-row varieties that we currently cultivate, there are 2 of these genes that are always, 2 kind of variations of these genes that are always together, okay? And that's the 6-row that's been selected over time, and that's what performs really well in the field. So what the goal of our study was to take some of these other genes that we know contribute to this trait and make combinations, pair those up, and see if they do any better. And what we found from our study is that some of these problems associated with 6-row, so grain uniformity, as well as those number of branches, were improved. So that is just an insight on maybe we can make some of these novel combinations, and in certain contexts they might perform slightly better.
As well.
Well, yeah, the TCD still is a fantastic piece of news for everybody. Not only people working on barley, but in the plant science community in Dundee as a whole. And absolutely, so the idea behind a portion of that money in the Tay City deals is for the International Barley Hub, or the IBH, and the whole major rationale behind that is to have an organization where you're doing the fundamental science right straight through to the industrial application. And so it's the whole idea behind it is to smooth that translational pipeline